So we've got two really special local guests, practitioners, here with us for this morning presentation. We've got Dr. Sarah Love and Dr. Wendy Abraham. Both of them are naturopaths, practicing naturopaths from Portland area here. Both received their professional training from our local naturopathic college, National College of Naturopathic Medicine here in Portland. Sarah has had a couple of different, she's kind of in a transition place right now. She's recently just finished, last week, I think you... Yesterday, this week, working as a population health specialist with Providence, their medical group there. And now she is just embarking on another really major project. She is the policy coordinator for CCO Oregon. So she's got a full plate ahead of her to be working on. I don't know if the power plate's probably going to help you with that, I think.
Definitely.
Yeah, so good. So we're looking forward to having her part of the presentation. Then we have Dr. Wendy Abraham, who is practicing naturopath, has been working in that field since around 2003, where she finished her degree there. She has in private practice at Sherwood Natural Medicine and Reflexology, which is different than what you have in the information originally provided on the website. And she's also the secretary for the Oregon Association for Naturopathic Physicians here in the area. So, but my connections with Wendy go back to about 2006 or so, I think it was. We were trying to figure this out. Wendy is the first person here on the West Coast to offer PCRM's, Physician Committees for Responsible Medicine's, Cancer Project program here. And in the West, I think you were the first one, at least in Oregon, definitely in Oregon. And so we had the real privilege at Adventist Medical Center to work with her to offer those programs for a number of years before her family expanded and she had to go to another, do something different with her evenings. But we had some really wonderful times working with patients on cancer prevention and things that they could do to help minimize their risk and promote good nutrition for cancer treatment as well. So they have a really fascinating topic, the gut flora as a forgotten organ and how plants can restore its function. So they're going to do a little tag team here, and we're going to start with Sarah.
Okay. Thank you so much for having me talk today. It's very exciting to be here talking with a lot of professionals who are very interested in food as much as I am. Kind of just a little brief synopsis of how I got into kind of food and nutrition, how that works is I remember, growing up and reading, growing up in Oklahoma, total tomboy, reading Ramona Quimby books because Ramona was awesome. And she lived in this great place called Oregon, and I got to learn all about Mount Hood and things like that. And one of my favorite memories in these books is when she's at the dinner table, she's like, I'm gonna say a bad word. I'm gonna say a bad word. And her family's looking at her like, okay. So she's like, guts, 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 guts. And everyone's just laughing, right? Because it's funny. So I just thought that was hysterical. So of course I was bound for Oregon to become a naturopath so I could learn all about guts. Great. I like starting with this quote. This is very new research, a lot of this that we're going to be talking about. You guys may have questions, and that's great. We have questions too. We do not have a lot of answers. So I'm just going to say that in the beginning. This is very new research. It's very exciting research. And there's a lot of things that we're thinking about, things that we're theorizing about, but we're still learning, and we're just at the infancy of really understanding and appreciating our gut and the microbiome. So this quote from Dr. Mealy out at UC San Francisco, bacteria within the gut are manipulative. There's a diversity of interest represented in the microbiome. Some are aligned with our undietary goals and others are not. So I think this is just kind of telling of where we're at in terms of really respecting and appreciating our gut bugs. So we're going to start by just kind of defining it as it's currently defined in the literature, looking at how diet can change the gut flora, looking at gut bacterial diversity and how that impacts health, talking briefly about antibiotics and our food-producing animals, as well as how we can promote gut diversity in the diet. So a couple of brief definitions. Sometimes you will see these used interchangeably in the literature, but just to make it clear when we're talking about microbiota versus the microbiome. The microbiota are the actual bacterial species that are in our gut, looking at large intestine, small intestine, where the actual genes that are in the bacteria, the microbiome. So just kind of thinking about those as two distinct things, but again, they're used interchangeably a lot in our common lexicon. So looking at the microbiome, so this is kind of housed everywhere in our body. We have bacteria on our skin, we have bacteria in our gut, lots of different places where we have bacteria, and we need them to be able to flourish and keep us healthy. So it's a lot of bacteria, bacteriophages, a little bit of fungi, protozoa, and viruses. They're living inside us and around us. And we have 10 times as many microbial cells as we do human cells. That's a lot of bacteria that we have to take care of. So I've heard some people say it's like, I'm really living to support my bacteria. And it's true, because they do outnumber us 10 to 1 cell-wise. So it's a huge population that we have to think about. And we don't see them, we just kind of have to trust that they're there, because they might manipulate us too, as we've discussed. So looking at the full gene content, again, the actual genes, there may be as many as 8 million unique microbial genes that are associated with this microbiome in healthy adults. So that is a lot of genes that we're thinking about. And Dr. Abraham is going to talk a lot more about the specifics of this and how it really does impact our health. But these are big numbers that we're just starting to discover and starting to realize that this does impact our health, and we need to learn more about it. So when compared to the human genes, so this is again just looking at the bacterial genes and the other small number of protozoa and viruses as well, the genetic contribution of this microbiome really does make a human superorganism, because there's 100 times greater of the genetic composition that's contributing to us is from this microbiome to our actual human genome. So that's pretty significant, and we really need to start paying attention to this and thinking about it. So here's just kind of some of the species that we have in our gut. I like to also put kind of gram-positive, gram-negative. When I'm thinking about antimicrobial prescribing in patients when I have to do it, I kind of want to know what bugs are going to be affected in the gut so I can think about how to maybe supplement those populations. So the Firmicutes, that is our kind of bacillus, that's our lactobacillus. That's also the species of the staph, as well as the Clostridium. The Bacteroides are gram-negatives, the Actinobacter, some more gram-positives. This is just kind of real basic. Of course, we've got multiple different classes under this, multiple different species under this. So these are just kind of our basic big three that are in the gut. So when we're thinking about how we're going to be changing our gut and changing the inhabitants that we have several different modifiable as well as non-modifiable ways to manipulate this. So of course the big one, the number one modifiable is diets. That's kind of what you always hear when we're talking about good health. What is the biggest one? It's always diet and lifestyle, diet and lifestyle, diet and lifestyle. Well, the same goes with this. Stress is another big one. Exercise, the more we exercise, the better it is for our gut bacteria. Lifestyle choices, smoking cessation is a big one in terms of that. Medications are big, especially antibiotics are the ones that everyone thinks about, but also in terms of other things that might maybe reduce some of our gut acid, making it to where other bacteria might get into the gut that we may not want. Some of the non-modifiable ones are birth. So I kind of put up here that 1/3 of births about in the US back in 2012 were C-sections. So we do know that babies born vaginally versus C-section do have different gut flora from birth. This is kind of something to think about. We can't change it, but I'm going into public policy, so these are things I think about. Early feeding, formula versus mother's milk, can also modify it, or non-changing it. And pregnancy, an interesting study that came out in Cell that I'm referencing here is in pregnancy, they took some, looking at what's happening in the gut flora, took some sterile mice and they did some fecal transplantation from women in their 3rd trimester of pregnancy, and they realized that these mice became insulin resistant as well as they gained more weight. So of course, when we're thinking 3rd trimester pregnancy, physiologically there's a lot of different things going on, and we could see how that gut flora may be contributing to that. But the insulin resistance was kind of interesting in that as well. We don't know, again, this is taking human feces and moving them into a mouse may not always be a great direct relationship, but it's definitely something we can think about in terms of our practice. So diet changes, this is where it really gets fun. This just came out this year, earlier in 2014 in Nature. So looking at exactly what is changing with diet. And we were realizing, we thought it might be, when authors were talking about when they were doing this study, they thought it might be like a week out, a couple weeks out, a month out. No, within hours of a diet change, they started seeing some differences in bacteria. When you think about the lifespan of a bacteria, it's pretty short and they can divide pretty rapidly. So it does kind of make sense that you would have some pretty drastic changes within hours. So they had a couple of volunteers that were doing this. One was assigned to very animal-based, kind of a higher fat, definitely lots of meats, eggs, dairy, and the other was a plant-based diet. And the gut microbiome responded immediately. The significant things that they found out were in the animal arm of that study, and that the gut, again, studying through the feces, not the number of microbes that break down the carbohydrates from the plants and were definitely decreased pretty quickly. And then they boosted levels of the bacteria that tolerate the bile so they can start being able to digest the fat a lot more effectively. So because plant polysaccharides can be pretty hard to digest, we need specific bacteria to do that, and if they're not needed, they're gonna die, which is pretty unfortunate. And bacteria, they like to fight and manipulate, so you can see how this would happen pretty quickly. One of the other things that they kind of briefly mentioned, and they're very clear to point out that this is not necessarily a direct relationship, but in other studies looking at patients with inflammatory bowel disease, they noticed that there was a huge increase in the abundance and the activity of this one particular organism, this Bifilo wadsworthia, on having a lot more in these IBD patients. They also noticed, this again a different study, but in this Nature study where the animal arm of the study, the people also had a lot of increase in this particular bacterial species. So they want to make it clear that there is maybe not a direct relationship, but it's definitely something interesting to note, and it's something we can start thinking about in terms of future research and future studies. I like that picture. Yes, bugs are everywhere. They're not all bad, though. We need to learn to love our bugs, the good ones anyway. There are some bad bugs that we have to take care of course. Okay, so this coming from another study, and again, using this word manipulating, which I'm finding more and more often, is that we're really starting to realize that our gut bugs do manipulate. So how are we going to deal with that? So microbes in the gastrointestinal tract are under selective pressure to manipulate their host, that's us, and they're manipulating our eating behavior. And this is something Dr. Abraham goes into a little bit as well in terms of the physiology of that. So they need to manipulate our eating behavior because what we eat is what they eat. So how are they doing this? They're thinking about two different strategies on this. So one, they're going to generate cravings for food that they specialize, or food that are going to suppress their competition, right? Of course, they want to grow bigger, they want to get their army, and they want to get everybody else out of the way. Second way is to induce this dysphoria until we eat foods that enhance their fitness. So this is where we can really start seeing the sugar cravings and cravings for, maybe some dairy cravings, things like this, because these are easily manipulated. So then we can also know this and we can fight against it when we're thinking about how we want to work with patients on their dietary changes, which will then alter their microbiotica. Okay, so let's talk about a couple of the functions of our gut bacteria. Again, we talked about competition. This is both good and bad. So because we do want the good ones to really be competing and taking over, we want them to be healthier so that they can kill the potential pathogens for the space and the resources. Big one is digestion of plant polysaccharides that would otherwise be unavailable. So those bacteria are very specialized, and as we've seen in that Nature study, they are going to be kind of not necessarily downregulated, they'll be killed off if they don't have any use. It's going to be promotion of monosaccharide absorption and triglyceride storage, priming of the immune effector cells, and then absorption of a lot of our ions. So when we're thinking about going on a plant-based diet, when we're talking with our patients, a lot of their questions are, well, how do I get calcium? How do I get iron? How do I get this and this? And it's not necessarily just about getting that in like one little line item on a food nutrition label. It's also about how we're going to be absorbing it, and having good gut bacteria primes the absorption for it. So if you've got a good gut bacteria, the absorption is going to be much better than just eating more of calcium in some other food and not absorbing it. Some other function, synthesis of some vitamins. Vitamin K is a big one. Vitamin B12 in smaller amounts, biotin, folic acid, and panthenate. It's also going to help with the biotransformation of our steroid hormones, bile acids, drugs, and dietary carcinogens. And the steroid hormones one I find really interesting, and it's one to where just kind of thinking about like fertility in patients. You can look at a patient, you start really transforming them to a plant-based diet, and you will see this, it's anecdotal in several different studies, and I've seen it in my own practice as well, is that patients can get pregnant a lot easier when you fix their diet a little bit. And maybe one reason is because our gut bacteria can help with those steroid hormones. Again, totally anecdotal, theoretical, but it would make sense. It's going to modulate the epithelial tight junctions and gut motility. A lot of what we talk about in school, as well as we work with patients about, is kind of this leaky gut type syndrome where the gap junctions are too big, so antigens or other proteins are able to get in. So functions of gut bacteria, good gut biome, will kind of help close those gaps. And then production of N-butyrate as the fuel source for the colonocytes. Looking at gut diversity, so looking at how many different species of bacteria that we have in a healthy gut. So the diversity of microbes within a given body can be defined as the number and abundance of distribution. And then looking at this gut diversity has been linked to several diseases that I'll kind of briefly go over, and then Dr. Abraham is going to get into a lot more detail on these. But one of the ones that I've already mentioned is that inflammatory bowel disease, and then obesity is going to be a big one with this. Celiac is another disease, and a lot of these, I'm just going to put this in here kind of for reference. You have access to the notes online, so you can read this a lot more detail. But looking at patients with celiac disease and studying their fecal matter and looking at the bacteria diversity that they have. And it's definitely very interesting. And then obesity is a big one, and type 2 diabetes is one that we're really starting to look at with this. So another one that just recently came out was looking at the low bacterial richness characterized by more marked overall adiposity, so a lot more fat that you'll see in patients, higher insulin resistance and dyslipidemia, and more pronounced inflammatory phenotype when compared to another individual with a higher bacterial richness in their gut. The obese individuals, among the, again, very low diversity in their gut, they're going to gain more weight over time as well. And this was actually a fairly large study that was published last year. So I put in a little bit with the immunity. Dr. Abraham's going to go into a little more details, but this is really kind of interesting when we start thinking about our gut diversity and what gut bugs can promote different immune responses in terms of allergies and how we can manage illnesses. And again, these slides are available to you on online. Okay, this is kind of a fun topic of mine. Antibiotics and food. Like I said, I grew up in Oklahoma. I grew up on a farm. We had cows and chickens and rabbits and cows and horses and all sorts of fun little animals that I took care of. So one of the things that we did not practice was one of the, you'll see it called in the literature stat therapy. So it's called subtherapeutic antibiotic therapy, and these are in the food-producing animals. So the FDA does have approved antibiotics for, and this is one of the actual indications, is growth promotion or increased feed efficiency in a herd or flock of animals to promote weight gain. And we don't know exactly why they're gaining weight with all of this subtherapeutic antibiotic dosing, but we do have theories that it might have to do with the gut bacteria. So maybe, in these animals that we're giving all these antibiotics to in their food, maybe that's why they might be gaining more weight more effectively. So starting very recently in 2009, the FDA started reporting on the use of antibiotics in these food-producing animals. So these are primarily the cows, the chickens, and the pigs, and really looking at what kind of and how many antibiotics that they're getting. Because over 29 million pounds of antibiotics are given annually to feed livestock, and this is between 2009 and 2011. So an independent person took it upon themselves to figure out exactly how many milligrams per kilogram was being used to get our meat and eggs. And he was using sources from the FDA report as well as the USDA report that documents annually how many pounds and kilograms of meat that we're getting from these different animals. And eggs are also included in this as well. So he calculated that for every kilogram of meat that we are eating or consuming or eggs, 300 milligrams of antibiotics went into that. So that's kind of a significant number. Here are just the classes of antibiotics that the FDA has approved for use in our livestock for growth promotion. So many, broad spectrum antibiotics that have been used. And a lot of these are also approved for use in humans as well. So the CDC came out kind of recently just thinking about this as a potential problem and seeing that food animals, they're as a reservoir for resistant pathogens and resistant mechanisms that can directly or indirectly result in resistant infections in humans. Resistant bacteria may be transmitted to humans through the foods we eat. Well, the CDC just came out with this in the last couple of years because they're starting to realize that this could be a concern for health. Some bacteria have become resistant to more than one type of antibiotic when we're doing cultures and sensitivities on our patients, whether it's urine infection or another infection. We really start wanting to know what antibiotics we can treat to treat that infection and not necessarily one that might be resistant to. So of course, because of this, we know that this is a huge problem in public health in that we're getting all these multi-drug resistant infections, hard to treat, they end up in the hospitals, and just kind of a rolling problem. Okay, so we've talked about antibiotics. Let's talk about prebiotics. So prebiotics are not bacteria. They're kind of the food for bacteria. It is an actual food ingredient. It has to have several different criteria for being defined as a prebiotic. Right? So it has to resist gastric acidity, so it has to be able to get from the mouth down into the gut. It has to be able to be hydrolyzed by enzymes that we have, and it has to be able to be absorbed in the upper GI tract. It can be fermented by our microflora as well, and fermented we'll get to in a little bit as well. And it needs to selectively stimulate the growth and or activity of intestinal bacteria potentially associated with health and well-being. So these prebiotics, because the bacteria like to eat them, sometimes pathogenic bacteria like to eat these as well. So we really want to make sure we have a good population of gut bugs. So looking a little bit at the history of prebiotics in the diet, because one of the big trends out there now is kind of this hunter-gatherer type diet and looking at, well, what did our hunter-gatherers eat, and maybe that would be good for us. So looking at these well-preserved coprolites, which is fossilized feces that they found in, this was in South America, they are suggesting that the dietary intake of inulin is one type of prebiotic, was about 135 grams a day. So right now, we are recommending 40 to 50 grams of fiber a day, and inulin is one of those fibers. And they were eating 135 grams of just this one fiber. Today, they figure we get around 5. So it's kind of a big difference in inulin consumption between ancient times and now. And inulin being a plant fiber is only in plant food. So I like it. It's also becoming, you'll start seeing it in a lot of processed foods now. One of the, you can find inulin and stevia now. I know. Inulin is great. Well, I talked about, here a couple slides back, I talked about being fermented by intestinal microflora. So when I'm working with patients and trying to increase their prebiotics after we've kind of gotten a good gut flora going, they've changed their diet, now we need to really kind of keep this going, because if we've got good bugs, we don't want them to manipulate us unless they're doing it in a good way. So we need to feed them well. So this fermentation, so when you're working with patients and you're working on getting them into more prebiotic foods, more inulin, and I'll give some good examples, make sure they do it slowly, because if they're going from 5 grams a day to 50 grams a day, that fermentation is not always a pleasant process for people. Okay, so here's some common foods. I talked specifically about inulin because that's the one I could find the research on in terms of what our hunter-gatherers were eating. Oligofructose, you'll often see it labeled as fructooligosaccharides, or FOS, and you see it in a lot of probiotic products. You see it added to yogurts. You see it added to a lot of processed foods. I'm looking specifically here at the inulin and the oligofructose in actual foods that we can buy. These are good whole foods. You can just go and buy them on a shelf, and trying to, maximize and gradually work up to getting more grams of that in our general diet a day, because again, this is going to be what's feeding our good gut bugs. So we need our gut bacteria to have good food. Bananas, asparagus are big ones. Chicory root, chicory coffee is kind of very popular in Louisiana. Inulin is water soluble, so you can get this out of like chicory root coffee, and stevia too, apparently. Globe artichokes, Jerusalem artichokes. Jerusalem artichokes, a big one, so people that are really going to town on Jerusalem artichokes may be feeling that. Leeks and onions, great source of the inulin and the oligofructose, as well as garlic. And of course, looking at these numbers, dried, just because you've taken out all the water, there's a much higher concentration of the inulin than there is in the actual raw product. And here are some other ones. Wheat, barley, rye, dandelion greens, great source of these prebiotic foods. Okay, so my last slide before I turn it over to Dr. Abraham. Again, with this, we are gonna just start bringing up so many more questions than we have answers for. It's a really exciting time in gut research. This is only the beginning. We're just learning about these bacteria living on us. We realize they're not invaders. They're beneficial colonizers, and we need to really appreciate and respect them. And we need to, because they're gonna care for us in the end. Okay, so I'm gonna turn it over. Where's...
Great. So thank you guys all for being here, and thanks for having us speak today. A continuation of the microbiome talk. So just a quick little question here. When I first started speaking, one bit of advice I got was to know your stuff. And they said there's 3 things about that. You need to know your stuff, know who you're stuffing, and know when they're stuffed. So, just a quick check-in. How many people are health professionals that are here looking for information more for themselves versus for patients? So, great. And how many people here looking for more patient, how to work with patient population? Awesome. And both. Yeah, that's fair enough. Good one. Oh, that's the pointer. All right. So the germs. There, as we talked about, there are good germs, bad germs, and this idea that we need to live in this sterile environment, I think, is really a big problem. When I was, several years ago, I was in an immunology class, and we talked about a study that was going on, and it was actually called the Lick Your Dog study. I couldn't find information on it, but it was about how kids that grew up on farms, around children, and children and animals together, that they had a healthier, more robust immune system and less allergies, and we're going to talk about that. One of the things I want to say kind of before, oh, that was the wrong clicker. One of my big goal here today is to really try to impress upon everyone just the importance and the gravity of the health of our guts. And as Dr. Love was saying, we are just starting to scratch the surface on what to do about it, how to take care of our gut. So most of what I'm talking about today is going to be, just different implications on the gut and mental health and the digestive system and so forth. So as we talked about and I'm just realizing my slides here are not advancing. So we'll just go with it and oh goodness. I shouldn't be trusted with one of these things. This is why my husband doesn't give me the remote. So we talked about, in different research I've seen that the microbial genes, anywhere from 100 to 150 to 1 of ours. So, we can have identical twins where we have the same genes, however, no two people share the same microbiome. And it's really important from approaching patients on an individual basis. This, I think, takes it to that next level. And it's important to know these genes, they encode proteins, and proteins help encode and set metabolism. This is just showing the stomach, from the mouth to anus, the different microbial populations, and where, and also like birth through death, the diversity. And a lot of this information is really for reference, because again, we're just sort of scratching the surface on the amount of information. Over the past, I think 3 weeks, I've been to several different webinar lectures on the microbiome. One was an hour-long, it was just on mental health. And then the other one was 3 hours on the microbiome and cancer. And so we've got about 20 to 30 minutes today to talk about all of it. So it's really, I tried to put a lot of info in there so you'd have information to take home and resources, but obviously because of the depth of this information, we can't really go into it in such detail. This is a really cool slide. We talk about the different microbiomes in children to toddlers, adults, with antibiotics, without antibiotics, breastfed, formula-fed, obese, malnutrition. And this is looking at just the different types and quantity and diversity of species. All right, we're just going to jump right into healthy poop and what makes healthy poop. So we talk about the pH of a good solid poo, and things that may increase the pH. So remember, an increase in pH is a more alkaline. And this kind of is confusing because people talk about, oh, you need to alkalinize, and alkalinize your cell. This is two separate things. So what they found is a high protein and low fiber diet can increase the pH. Dysbiosis, which we're going to talk a little bit more about. Hypochlorhydria, where there's not enough stomach acid to break down our food. An increased bile flow rate. These can all be associated with increased risk for colorectal cancer when we see that the increase in pH. Whereas a greater acidity, a lower pH in the colon has been more health promoting. Okay, so short chain fatty acids, these are fuel, this is fuel for the colon cells, and really we really want to sort of nourish the ability for our body and the gut bugs to produce the short chain fatty acids, because they are that fuel. They're generated in the colon from bacterial fermentation. So again, that population of the gut bugs that we want the guts, this is disorienting. that the colonocyte proliferation can help reverse colon atrophy, and the presence of butyrate at physiologic concentrations here enhances the growth of normal cells and inhibits malignant cell growth and helps with DNA repair. So pretty important in colon health. Here are some studies. This is one on the effect of fiber and the source of on short-chain fatty acids. And basically that fermentable fibers support the growth of indigenous intestinal bacteria. And these factors contribute to the prevention and growth of the toxins produced by C. diff. I'm going to attempt to, let me know if it's changing behind me, but I don't want to. Sorry. I want to be looking at the same thing you are, so I'm not having my back to you the whole time. Oh goodness, this is. So now this is, now it's advancing my slides here and not up there. Sorry. This is when I'd like to phone a friend. Excuse me? Are the slides downloadable? Yes, they are online. Mm-hmm. Well, it's moving here and not there. And what I see here.
Which one do you want? You want this fiber one or?
Yeah.
What if we need to put? Oh, yeah.
And
is that it? Is it that? No, I'm wondering if you switched off to the presentation.
sorry.
It might not be cued.
Oh, that might. Oh, this is nice. Okay, much better, thank you. Okay, so a little bit more on fiber. As we talked about before, fiber is really, really important. If there's one major take-home message from this, it's to really focus on fiber. And this study here, and I've bolded and underlined this, the low-grade inflammation part, and I don't expect everyone to be able to read that real quick, again, just for reference. But this shows that a strict vegetarian diet improves the risk factors associated with metabolic disease by modulating, so it did this via modulating the gut microbiota and reducing intestinal inflammation. There's some interesting information, if anyone hasn't already come across this. Jeff Leach from the American Gut Project, and he did this study where he, on himself, did no fiber for 10 days. And what they found was that the abundance of the Firmicutes, this one variety of bacteria, which is specifically a butyrate-producing Clostridia bacteria, was driven by the consumption and fermentation of plant fiber. And what they also found, which was fascinating, and I think this has kind of come up in the previous lecture about with smoking, but the consumption of plant fiber led to the exact same microbiota shift as smoking cessation, and which I thought was pretty cool. Along those lines, you know, and there's the whole, and I'm always so reluctant to say, you know, the paleo diet, because I think when people talk about the paleo diet, it's this assumption that this is how the Paleolithic folks ate at the time, and I think that's a big misnomer, because I don't think, you know, they had, you know, constant access to bison, and it was not as meat animal dense as what sort of, when someone says paleo, you just automatically think they're just eating a ton of, you know, bacon and, you know, food. The study here shows, this was this breakdown of the microbiota of, it's probably hard to see this, the one on your left here is where the diet had lots of meat, and meat and fat with lots and lots of plants and fiber. The one on the right was lots of meat and fat and no plant fiber. And so the biggest thing here is, the thing that really made the biggest difference was the addition of the plant fiber. And it's funny because one of the talks I was at recently, and it was on the microbiota and cancer, and the whole talk we talked about, it was really about the gut, the diversity and the gut. And then one of the last slides they mentioned, and it turns out that with all this antibiotic resistance, it looks like a paleo-like diet is the way to, or paleo lifestyle, it was worded very strange, is really our only hope, which seemed very odd to me, because I was like, well, everything you just talked about really was not saying that. So I went back and I found the research, and it calls out in the abstract the plant, it was something to the effect of including a lot of plant, vegetables and fruits is the piece of the paleo whole food piece of it that was really important. So I think we have to be really careful when we start throwing around this idea of paleo or eating like our ancestors from that perspective. So again, this is a little review again about the factors affecting good bacteria. And the thing I want to talk about here is the sterile environments. This whole, wiping down our food carts and wiping down, antimicrobial hand wash. It's really affecting our total microbiotic diversity, and in creating this impoverished microbial community, and is having, I think, negative impacts on our immune system. And I was telling this story about my daughter who's 4 is in preschool, and they have their bottle of antimicrobial hand wash, and it's called Germy Juice. And after, before everything, they get to take turns, passing out the Germy Juice. And I'm always telling her, don't use that. Just wash your hands. You don't need to, disinfect. No one's going to surgery here. Just eating food. So dysbiosis is a term of when you have an imbalance of gut bugs. So we got the good, the bad, and the ugly. So the good are the ones that are going to do all those things that Dr. Love talked about as far as, in creating that good environment. The bad are probably the ones that are not necessarily termed pathogenic, but just by having a presence and an imbalanced presence, they crowd out the room for the good guys. And then, of course, the ugly are the ones that are known to be to cause disease and pathogens. And here's the short list of all these different symptoms that are associated with a dysbiosis. And it's funny because from a naturopathic perspective, the gut has been the foundation of our of health, and it's kind of where we always start with the gut. And so it's great now to see that research and things are catching up now that they've identified cracked the code to the human genome. Chronic dysbiosis can lower the levels of the beneficial short-chain fatty acids that we talked about and alter the bacterial metabolic activity, thereby increasing the risk for carcinogenesis, cancer-causing hormone imbalance, and inflammation. Here's a list of the common GI conditions that are associated with a dysbiosis. And a lot of these things we already know about, and we would think, that makes sense that, you'd change the gut bugs, and of course you'd have symptoms of constipation, diarrhea, inflammation. The things that we don't always think about being associated with dysbiosis, obesity, metabolic syndrome, anxiety, depression. So mental health is a huge area where they're now finding that this dysbiosis is connected to an imbalance of the microbiome. This is a new term that I learned when I did this last talk, was the psychobiotics. And this is a live organism, so example, a probiotic that when ingested in large amounts, in adequate amounts, has a positive health promoting effect for people suffering from mental health issues. And this has been shown to be effective for people suffering from depression, anxiety, OCD, trichotillomania, where people kind of pull out hair in chunks. Just giving probiotics has helped reverse that. They found that there's this gut-brain axis. So, a lot of people talk about those gut feelings and kind of going with the gut. I think it's going to be coming out a lot more, sort of really what this, what's driving this. And basically, we need a proper, healthy gut environment, and that helps with our actual brain development. And there's the vagus nerve, which drives this communication between the gut and the brain, and the gut microbes, and this is one of the ways that they manipulate us, is through, they're able to send signals. For example, Candida has been known to actually create neurotransmitters that go to the brain and, tell you, kind of whisper in your ear, each other, and, because it feeds them, and that's what they live on. It's the whole, survival of the fittest. So I kind of question if our gut is really, they say it's our second brain, and, I'm not convinced that it's not really our first brain, because again, more genes, more bacteria, more cells. And here's just some evidence that, maybe suggests that we need to look at that. So there's actually more nerves in our digestive tract than there are in our spinal cord. Our gut produces the same neurotransmitters that are found in the brains, like serotonin, for example, which regulates the mood, sleep cycle, pain, to name a few. Before we jump out of the mental health, there was this study on personality and the microbiome, and they took two groups of mice. One was a group of timid mice, and the other was a group of aggressive mice. And they took stool samples and did something, I don't know what, to sterilize there. They sort of did a wash of the intestines, and then they switched. They basically did a fecal transplant. They gave the timid mice aggressive mice poo, aggressive mice timid mice poo, and lo and behold, the timid mice became aggressive and the aggressive mice became timid. Like, just by changing their poo, which I think is pretty impressive, and so, whether that translates into humans, and I wouldn't recommend anyone going to, find someone that you think has a nice personality and taking on any homes, things. But just the fact that that has the ability to have such an impact, I think, again, kind of leads credence to what we're talking about here. I'm just going to check in on time. So this is about the microbiome, and I don't know why that's quite so small. But again, some studies talking about changes in the gut microbiota and how this affected the toll-like receptors, and how this can induce acute intestinal inflammation, and how that's linked to inflammatory bowel disease. Basically, what happens here is that you have a shift in the healthy symbiotic microbiota, and that shifts into this dysbiosis, which in turn changes the host's tolerance, and as we talked about, causes some leaky gut in the blanking on those tight junctions, and promotes Th17 differentiation, which then causes some of the interleukins that are pro-inflammatory to start producing pro-inflammatory signals. Here on the left, we have sort of a healthy homeostasis situation, and then on the right, more of the dysbiosis, creating a chronic inflammatory pathways via these toll-like receptors and interleukins that modulate inflammation. So there's a lot of questions around, probiotics, yogurt, all the, good gut bugs, belly bugs, as we call them in my house. There was a double-blind placebo-controlled study that studying basically the effect on probiotics and allergies in children. And it was studied with 454 mother-infant pairs who took a daily dose of probiotics. So they took about 10 billion of a specific strain, and then they followed the kids through the first 6 months of their life. Or they gave them for those first 6 months, and the babies that were then assessed at 2 years of age, and it was found that the group, the probiotic, that were taking the probiotics significantly reduced their chance of becoming allergic to common allergens such as pollens, cats, dogs, cow's milk, and eggs. It was cut in half. And the children of the mothers that were taking the probiotics had a 60% reduction of developing atopic eczema. So this is pretty useful information for those working in, pediatrics, working with pregnant moms, and trying, from a prevention perspective, getting into some prevention of allergies. That's a huge, huge issue for kiddos. This is interesting. How many people, and I saw this kind of go around Facebook a while back, of this link between heart disease with atherosclerosis and carnitine. Anybody come across anyone's? This is really interesting, and again shows this shift and the importance of the microbiota. This, they made this link between, and people are eating these high meat diets, it's too much carnitine, and carnitine is driving this link with atherosclerosis and this meat and cheese consumption. And what they found was it isn't actually the carnitine, and not necessarily specifically the meat and cheese per se, but actually the specific gut bugs that are there when someone is eating a lot of meat and cheese. So you have people that are eating a lot of meat and cheese, the shift in the gut bugs happens, and those gut bugs in turn are able to produce this trimethylamine, which gets converted in the liver to the TMAO. This substance is what encourages the atherosclerosis and interferes with liver enzymes that synthesize bile acids and so forth. And this TMAO synthesis requires the specific microbes that digest carnitine. And so it was interesting because they started to figure, well, is it the carnitine, is it the meat? And what they did was they gave really high doses of carnitine to vegetarians to see if they would still get this rise in the TMAO, and they didn't. And so they then found out that, it's the gut bugs, and it's the presence of the specific gut bugs that are populating the typical gut of a carnivore, basically. So that was pretty cool. So they didn't see the atherosclerosis connection in the plant-based diets when given, the same high dose of carnitine. This came from when I think this will be on the online. I updated this a little bit last night. This is a study where they looked at the effect of antibiotics and disappearing microbes. So this constant, impact of having antibiotics and linked to breast and prostate cancer. And they found, although they said it's a non-causal relationship on one hand, that they observed a dose-dependent increase in breast cancer risk and associated with antibiotic exposure when that's happened up to the past 15 years. So basically, if you've taken antibiotics in the past 15 years, the risk of breast cancer goes up. Again, they said it's, there's a lot of variables, a lot of factors there, so they can't necessarily suggest a non-causal relationship. That said, the number of antibiotic prescriptions over that time was significantly associated with an increased risk of prostate cancer. So again, a lot of this is just really trying to really hit home that the impact when we do things that shift our gut, the impact that it can have on our health. This slide is for people that kind of want to geek out on some immunology. There. And for time, we're just going to move through that. And this is talking a little bit more about the cytokine interactions and the microbiota and how that plays a role in inflammation and also cancer. I wanted to add this. Soy has always been a big, I would call it the great debate of to do soy, to not do soy with estrogen, and when they look at different cultures and why. Why is there less breast cancer in Asia when they have a lot of soy consumption, but they come here, and this whole idea of, eating a westernized diet and what's really at the basis of that. This was really fascinating. It turns out that the conversion of isoflavones requires a proper microbiota. And I think it's even more interesting that the breast milk, so you have a mother who's consuming isoflavones, soy, when those child acquires the bacteria through breast milk, it helps to make a microbiome that's capable then of metabolizing that. So we see that if it's beginning in infancy, that gut microbiota, it basically helps to be able to then later in life convert and make that conversion of isoflavones. It's pretty cool, isn't it? It's like, and this is looking a little bit same thing as but with prostate cancer, and looking at how the role of intestinal bacteria can be chemoproductive with respect to soy isoflavones. People often talk, about autism, and there is a link with autism and the microbiome. And as with most of these conditions, it's definitely multifactorial. You can't just, give some probiotics or eat a bunch of inulin and expect that it's going to take care of everything. But it is definitely a piece of, this chronic health, the puzzle. And there are some studies that show that children with autism lack one of the healthy fermenters, this Prebiotics organism. And interestingly, the study that Dr. Love had talked about with the vegetarian and the meat, where they switched and you saw how fast the microbiota was impacted, that the group, the vegetarian arm, those subjects, when they went to the animal consumption, that Prebiotics organism went down too. So it was there in the vegetarian groups prior to doing the animal ingestion, and then back down. I should have made this disclaimer in the beginning that I like to, when I start doing research, I tend to head down random rabbit holes, I call them, and I just get really excited about, different connections. And so this is one of those rabbit holes where looking at a potential connection between a dysbiosis, autism, and I got a little bit into Tylenol here, and defective genetic pathways. So for those interested, you can take a look here. It's pretty interesting and kind of ties everything together. But I want to get through so we have some time for questions. This sort of a new term, the hollow biome theory. And this is kind of just taking all of this information, kind of the survival of the fittest, this idea that we're carrying around this whole super organism, and you are what you eat, you are what you don't eliminate, you are what you don't detoxify. These are all different things that I've heard through going through school. You are what your microbiome is. But basically it's this idea that all animals and plants have this symbiotic relationship with microorganisms. We need the gut bugs, they need us. And that this population, each population sort of has its own symbiote relationship. And the interesting thing is that the gut bugs, as we've seen, they change and they can become more adapted to the environment faster than we can. And so I really think they're driving so much as far as natural selection goes. It sort of came based on all this evidence that the holobiome within its hologenome should be considered as the unit of natural selection and evolution. So I feel like I just threw a ton of information at you guys really fast, and I'm probably at that stuffed limit. But I just think this whole area is very fascinating. Again, we're just scratching the surface. And to close, this is one of my good friends and colleagues. The road to optimal health is paved with good intestines. So with that, we're going to adjourn for lunch after questions and feed our gut, all that good stuff. But any questions?
You mentioned the term psychobiotics, and I'm just wondering if the literature supports any supplementation with a particular probiotic.
So the questions as far as the psychobiotics and any particular supplementation, they don't talk about specific types or strains. And I found in practice that a lot of times you just need a variety of strains when it comes to supplementation, that our bodies, we're just, again, at that surface of what, specific strains, we're not testing So really just a diverse, and some people it's a pretty high dose of probiotics.
I'm just wondering, you had mentioned something about pregnancy and probiotics. Is that something you're actually recommending for your pregnant women? And is there a point during the pregnancy that you start the probiotics or stop? Or, I mean, what do you usually recommend?
I recommend probiotics for most people, and definitely during pregnancy and also for the newborn.
Do you have a specific brand?
Though I probably use two different. I use a human. It's a human strains. So human strains. I use an HMF or HLC, human microflora. It's suitable for vegans.
Two simple questions for you. First one is, nature abhors a vacuum. What probiotics occupy the space inside of the stomach? And are there any that have been identified? And then the more difficult question, while you were preparing this week, 3 new studies came out, one from Oklahoma, identifying 500 new probiotics with 8 billion new microbiomes, and a second one out of Chicago, identifying one of the species of Clostridium with deactivating the anaphylactic peanut reaction in children. And this is the question about evidence base. Loudon Douglas was the last scientist in 1911 to look at the probiotics until the 80s. We're probably talking about thousands of populations. When we link one microbe with a disease condition, are we not taking a view from the stadium of the broad impact of dysbiosis versus the singular heroism of a single microbe?
Yeah, thank you. I think, I'm not sure if your first question about the stomach, there was, this slide here talks about the different bacteria and the quantities found in the stomach. And it's interesting, the whole H. pylori, is a bacteria often associated with different conditions, ulcers in the stomach. And, there's sort of the studies on, well, what do you use to eradicate that? You kill off the H. pylori, you're killing off the bacteria in the gut. So it's a bit of a conundrum. And I would say, as with anything, when we start just pointing in a real mechanistic way to say, oh, you need lactobacillus, because there's so many bacteria, there's no way to test for all the bacteria. Hold on.
And again, yes, I did see that those 2 studies came out from Oklahoma and Chicago this week. And again, like I was mentioning, there's some great information out there, and there's a lot of observations that we've had in terms of being able to connect one bacterial species with one disease. Nobody's willing to make that leap now because we have way too many species to choose from.
I had a question about the long-term use of acid-reducing medications and gut flora. If you could comment on that.
Yeah, acid-reducing medications, whether it's the proton pump inhibitors or the H2 blockers, those are kind of the 2 big ones. We don't know. Yes, we do think that there's definitely some relationships. When you look at calcium absorption in long-term proton pump inhibitor patients, we know that it's lower. So yes, that is definitely an area of interest and one that is being looked at, but in terms of definitive studies, not yet. But we do know that there are some relationships.
And I think that does contribute to a leaky gut in the stomach. So I think we'll start to see some issues there as well.
This is a 2-part question. The first part's kind of serious, the second part's not so serious. It's in reference to the mouse study that you referred to, where there's an exchange of their flora and their behavior then altered. Is there any evidence or any hypothesis or any legitimacy to the idea that the exchange of fluids in romantic intimacy could affect the gut flora in any way for an individual? And if so, can this explain a change in behavior after entering into a romantic relationship?
Those are great questions. And I have no idea.
It's a cautious yes, because we do know that when you're looking at fecal transplants in terms of C. difficile helping, we always, when they're looking at transplant, they always look at, who's living in the same house for several reasons, one of which being that they are in close proximity, they are sharing a lot of different, through touch, through romantic relationships, through diet, they are sharing a lot of those bacteria. So yes, but we don't know. So how's that?
I'm a nurse in a hospital where everyone is on a PPI or an H2 blocker, and most people are on one, if not two, IV antibiotics. Currently, we give Culturelle to most of those patients. But I'm curious if there's any research that shows the efficacy of taking that while on the antibiotics, and if there's anything else that I can do to advocate for my patients.
I do know that in terms of ICU patients, they're on H2 blockers to help with stress reduction. So there is definite indication for that. In terms of other things, because I used to work in an ICU, I know there's a lot of doctors advocating for that in the hospital population for many reasons, just because of antibiotic-associated diarrhea and infections. But I don't know that there's been, and there has been a lot of research out there in terms of specific antibiotics and looking at what probiotics we can supplement in those patients. The research is out there. I'm not as familiar with that, but it is there.
I would just add, a lot of times it's the, especially with oral antibiotics, because you're getting that direct impact on the gut. You see less, I'm sure there's some, especially when they're on, IV antibiotics, Q4 hours, but it doesn't have as great of an impact on the gut when it's intravenous antibiotic versus oral routes of administration. Typically with an oral route of administration, you want to make sure to give the probiotic at least 2 hours before or 2 hours after. Otherwise, it's going to just, the antibiotic will kill off the probiotic as well.
Hi, I'm really interested in the B12 synthesis in the gut, and I would really love to not have to deal with the supplementation of B12. It looks logically to advocate a plant-based diet and say, but you got to take this pill, to me is just really frustrating. And I went to a doctor who said, you've got the intrinsic factor, you're good to go. How can that be tested and easily for people? I think I've said enough.
Yes, our gut bacteria do work in the synthesis of active B12. However, we don't know that that is going to be enough without having it in the diet. So I do not advocate for just stopping B12 supplementation until we know more information. That said, as much as we would like to get people off of a lot of these different things, I advocate for vitamin D as well. So, that's because we're definitely not getting enough of that, anybody in the Pacific Northwest. So, and you can also test for levels of active vitamin B12. But yes, and same with vitamin K. Yes, our gut is synthesizing the active form. Does that mean I want people to stop eating dark leafy greens? Absolutely not. So I think that until we learn more, until we learn exactly how much our gut can make. We do know that when patients or when individuals go on a very strict vegan diet, they have about 5 years before they actually have to start supplementing B12 and become really symptomatically low. So there's a little bit in there chugging around, but I still can't advocate for stopping B12 supplementation in vegans.
Hi. You talked a little bit about bad bugs taking over the gut and some diseases that can take place. I wanted to know what your thoughts were about limiting fermentable carbohydrates in the gut as a therapy.
Like the Fodmaps diet?
Yes.
Yes, okay. I was waiting for the Fodmaps question. I know, I totally realized this morning, I didn't put a slide up on Fodmaps. Okay, so for those, who actually is familiar with the Fodmaps diet? So a fair number, this is great. Okay, so Fodmaps Diet is looking at the fermentable oligopolysaccharides because sometimes they can cause a lot of GI problems. And in a lot of patients, removing all of those fermentable oligosaccharides can definitely help with their symptoms. I'm, of course, was talking about inulin, which is a fermentable polysaccharide, and looking at gut bugs. If you have patients that are doing really well on a Fodmaps diet and have a reduction of symptoms, that's fantastic, because I know people who have done fantastic on their Fodmaps diet. And the last thing I want to do is say, oh, hey, have a Jerusalem artichoke, you'll feel like hell, you know? So it's just not a good thing. I wish we definitely need to, I think the Fodmaps diet is great for a lot of different patients. I think it can be very well prescribed and monitored in patients. In terms of other patients, you know, maybe at the same time when you're looking at that Fodmaps diet, you can really focus on maybe just kind of tinkering a few, you know, bites here and there of some fermentable carbohydrates and seeing what works for them. But that would really have to be on a case-by-case basis, because patients who are doing well on a Fodmaps diet, they are having such a great joy in the reduction of symptoms that giving them something that you know may aggravate symptoms is going to be a little hard.
I think the only other thing I was going to add to was Dr. Bailey's comment about the one strain of bacteria and peanut allergies. There has been some interesting research on, and it's directly involved with the gut bacteria. It shows that you know, when we have allergies, we've lost tolerance. Our bodies have lost tolerance for whatever substance that is, and there was a study, Dr. Nigel Plummer has done a lot of this work, and he was in this seminar, he was talking about how they had done a study where they gave, you know, they took whatever number of kids that were highly allergic to peanuts, and they gave very, very, very small amounts administered with a probiotic, and that probiotic shifted the I don't know if it was a toll-like receptor or what going on in the gut. And when they were able to do that, they were able to then, the kids had to eat, it was like 14 peanuts a day to maintain tolerance. But they were able to eat peanuts and come in contact with it, but they had to maintain that to maintain the tolerance with the probiotic. And so there's a whole area of immunology and research going on around that. Same thing with rheumatoid arthritis and different, you know, joints, taking that with the probiotic. So thank you all for your great questions.